Okay, folks, now we're going to focus on Luminex-based analysis. But before we get into Luminex, I just wanted to discuss the different levels of quality that can be involved in these multiplex assays. Um, so a lot of these companies do a broad range of targets, but you need to be you need to consider the level of validation that some of these assays have underwent. For instance, the MSD platform has 3 or 4 levels of different levels of validation. They do have a lot of markers that can be used for fundamental research, um, and they also have another range of markers that can be used for in a clinical setting, where basically they've been able to clinically validate the markers of interest, so that they can be used in a clinical setting for prognosis, diagnosis, etc. Then they'll also have different levels of sensitivity as well. So these are all things you need to consider when looking at multiplex in terms of how you're going to use it and what level of quality is required for your outcome. Okay. A lot of these companies that do multiplex assays have a lot of assays that are not clinically validated, and if you're doing human studies, you kind of have to be careful with that. Okay. So in terms of the Luminex-based systems, I mean, traditionally, the ELISAs that we've talked about before, generally speaking, involve some degree of immobilization of either the antigen or the antibody on a solid matrix or a plate. For instance, in this case, when we have an ELISA that wants to quantify insulin present in a serum sample, we will immobilize a anti-insulin antibody capture antibody on the plate. But the difference with Luminex is that what they do is they immobilize, if you like, the antibodies on beads. Okay, these colored beads. And essentially what happens is they can then mix the beads with a sample in a fluidic-based system. So then when you add the sample, the target, in this case insulin, will be able to bind with the antibodies that are attached to the beads. I'm moving on then to the multiplex approach. So again, in your standard ELISA, you'll have different capture antibodies or different antigen-specific antibodies on the same well or reaction space. As you can see here, for instance, this assay wanted to look at glycemic control and appetite, so they looked at insulin, GLP-1, GIP, and anti-glucagon. And what that enables the individual to do is to add one sample, and within that one sample, you can measure 4 different markers at the same time, and more markers depending on the number of different markers that you have. But that would be your standard approach from an ELISA perspective. But from a Luminex approach, what you would have are different microspheres or beads that have a different color profile. Okay, so for instance, here you can see this particular bead has anti-insulin attached to it, antibody. This might have anti-GIP. This might have anti-GLP-1. This might have anti-glucagon. Now, the bigger question here is, how is the machine going to know what antibody is bound to what bead in terms of quantification? Well, the way the Luminex system is able to do that is it gives each one of these microspheres or beads a unique color. Okay, so the fact that the user knows that insulin antibody is bound to the purple bead, all they need to do then is tell the computer, when you detect a purple bead, you will be measuring insulin, and so on. So that's how the Luminex-based system works on a very basic level. And how is it able to determine what color the bead is? It uses a series of lasers. Okay. And just to be mindful that each bead that has a job of detecting a target has a unique cocktail of different dyes. So basically, every bead that's directed against a target will have its own unique color, and that unique color enables each bead to be given its own ID, if you like. Um, so each bead can have a specific target antibody attached. For instance, the red bead might have insulin antibody attached, which will enable to quantify insulin in the sample. Likewise, you might have a green bead that has anti-glucagon antibody on it, which again will enable us to quantify glucagon within the sample. And of course, the in terms of the Luminex technologies, the benefit is that they have the ability to create a vast, diverse range of different colors for the beads. And of course, the more different colors you can use for the beads, then the more targets you can investigate. And I suppose the key for this technology is the ability to be able to distinguish one color bead from another color bead to enable to measure different targets reliably. And in this technology, there is certainly one device that enables the machine to be able to detect up to 500 different types of target, which is incredibly impressive. Um, and it enables a number of analytes to be measured simultaneously in the one sample. So what it's saying here is that if you design it properly, you have the potential to quantify up to 5 different targets in the one sample. Now that may not be realistic from a clinical perspective, but certainly a research perspective, if you're doing sort of forms of screening to look at changes that take place in a cell, for instance, then that might be a useful approach. So here's just a sort of a visual example, I suppose, that, here you can see each one of these beads, as an example, of course, each one of them has a unique color profile, which means each one of those beads is able to be used to measure a specific target. And again, how are they able to distinguish one bead from another? Because you can see, the colors are different, but they're quite similar at the same time. And the way they do this is through the use of lasers, okay? So they use one laser to identify the actual bead, okay? So they use a red laser to ID the color profile of a bead. And of course, if you're able to identify the color profile of the bead, you'll be able to tell what target that bead was allocated to measure. But then, of course, once you identify the bead, the other thing we need to do then is to quantify how much of the target was actually bound to the antibody on that bead. And we do that through the use of a green laser, okay? And a green laser will be able to measure the amount of target that's bound to the antibody on that bead. So this sort of approach that I'm talking about now, there are other, I suppose, different approaches in the Luminex technology, but the approach I'm talking about now is almost very, very similar to a flow cytometry, if you're familiar with that. So the Luminex concept is you get a bead, you give it a unique color, then you attach the capture antibody of interest. For instance, if it was insulin you were interested in, you would attach a capture antibody that's anti-insulin. Okay. Next thing you would do then is expose this bead with the capture antibody to the sample. It might be blood, it might be urine, whatever it is. And if the target is present in that sample, it will bind to the capture antibody. The next thing you then need to do is attach a detection antibody. Well, the detection antibody will have a biotin label on it, and that will then, the detection antibody will specifically bind to the other side of the target of interest. And then you would subsequently add streptavidin which will attach to the biotin on the detection antibody, and that will subsequently enable you to quantify the amount of target bind based on the fact that streptavidin will give off, when it's stimulated, will give a fluorescent response. Okay, so how do you get the result? The first thing we do is we need to identify the bead in question. Okay, so using the red laser, we're able to detect what bead is being identified. So, for example, you want to identify the bead which you know is labeled with anti-insulin antibody. Once you've identified the bead that we know is destined to target or to quantify insulin, we now need to quantify how much insulin actually bound to that bead. And we do that by measuring the amount of target that's bound to the antibody using the green laser. And of course, we're able to quantify the amount of target based on the fact that when stimulated with a green laser, it will cause a color change and will enable us to detect the streptavidin present. So at this stage, you've identified the bead in question, and secondly, you've quantified the amount of target that's bound to the detection antibody the capture antibody on that bead. Okay, so this is sort of approach. You expose the sample with the antibody conjugated beads, you incubate it, and of course, if target's present in the sample, it attaches to the capture antibody. Then we subsequently add biotinylated detection antibody, which will bind to the other side of the target of interest, incubate that for a while, and of course, if target's there, the detection antibody will attach. And then subsequently we add streptavidin, and it will subsequently bind to the biotin that's present on the detection antibody. And then subsequently using the machines, through the stimulation of using the green laser, you'll be able to detect the amount of target that's present. So that's sort of the basic concept. And of course, like most things, there are caveats. The Luminex-based system, generally speaking, you may come across the use of what I've talked about so far, which would be the flow cytometric-based detection, where you interrogate the beads with a red laser and a green laser, and you'll be able to identify the bead and quantify the target of interest. But of course, there's other technologies that they have as well, which are based on LED-based stimulation, but the underpinning of it is predominantly the use of magnetic beads. And using a magnet, then the machine can subsequently organize the beads into a monolayer, as you can see here, into a very clear monolayer, and that monolayer of well organized beads will then be stimulated with the red LED and the green LED, and it will subsequently be able to quantify the amount of target present. It's giving you the same answer, just using a different type of approach. But the throughput for this sort of magnetic technology is limited to about 50, whereas the flow cytometry can enable you. This flow cytometry detection system will enable you to reach up to 500 different targets. Okay, so this is basically the same sort of approach, and then of course you stimulate the both. So there are 3 different machines. There are other machines for the Luminex, and I think it's important as well to highlight that the Luminex-based system can detect proteins, etc., of interest through the use of detection antibodies. But Luminex also has the capability to detect genomic targets as well, through the use of nucleotide hybridization. And that's an emerging area of interest as well. But yeah, I mean, there are different machines that are present. The MagPix, which is based on a magnetic-based technology, and then the other 2, which are based on fluorescent-based approaches. So yeah, they're the sort of 3 machines that we're going to talk about now. But As I say, there are other machines that aren't talked about here, that can also be used for which are predominantly more focused on the genomic approaches for Luminex's technologies. So we're just going to go through a few videos to reiterate what I've just said and to give you an insight into Luminex. So we'll just go now to this.
Luminex XMAP technology makes up the basis of Luminex's core business. This flexible technology is centered on the use of Luminex XMAP microspheres.
XMAP was originally an acronym, meaning multi-analyte profiling, where you solve for X. In other words, analyzing data from an assay, checking for multiple analytes in a single test.
The surface of the microsphere is covered with carboxyl groups, which offer multiple chemical attachment sites for biological capture molecules. The attachment of molecules to the microsphere is called coupling. Carboxyl groups on the bead are reactive, allowing bonding to both nucleic acids and proteins. Because carboxyl groups are so reactive, it is a versatile and easy-to-use chemistry. With the bead coupled to the desired capture molecule, it can now bind with a specific target. Using a reporter molecule with the target makes identification of that target possible.
For example, beads 12, 35, and 42 have specific dye concentrations that are detected by Luminex instruments and used to identify each bead. Additionally, each bead has a single target it can bind with, which is determined by the capture molecule that is attached to the bead. For example, either X, Y, or Z, respectively. So if target X is present in the patient sample, B12 will bind to the target. The target is then bound to a reporter molecule that the Luminex instrument also detects, associating it with B12 and identifying that the patient has disease X. The other beads are still red, but since targets Y and Z are not present, no reporter will be detected on those beads. That is how XMAP technology works.
The analytes of interest are commonly nucleic acids or proteins. Following are examples of specific uses for nucleic acids and proteins.
Nucleic acids, particularly oligonucleotides, are common analytes of interest that can be coupled to microspheres. A nucleic acid sequence specific to the target is attached to the carboxyl group using an amine linker. The amine linker positions the capture probe away from the microsphere's surface and facilitates binding to the target. When the sample is added to this reaction, the nucleic acid sequence on the bead binds to its complementary nucleic acid in the sample, if present. This is called hybridization. Biotin is incorporated into the target to provide a binding site for the reporter molecule.
Proteins are another common analyte of interest that can be coupled to microspheres via the primary amine groups naturally present in proteins. Coupled proteins can be used as capture molecules to bind specific protein targets. Protein targets can be directly or indirectly identified via binding interactions with capture proteins attached to the microspheres. These binding reactions occur between various proteins, including antibodies, antigens, receptors, ligands, and enzymes.
Luminex currently has 3 XMAP instruments based on either flow cytometry or fluorescent imaging technologies. These instruments are used to analyze assay runs and to provide data on analyte presence.
Luminex XMAP technology is based on the use of Luminex microspheres. The reactive surface chemistry of the microspheres provides a flexible platform for the attachment and detection of multiple types of biological molecules. Luminex's wide range of XMAP products allows for a customizable end-user experience. Of Luminex's technology utilizes microspheres, also called beads. Microspheres are small, spherical particles with diameters that are measured in the micrometer range, thousandths of a millimeter. They are typically not visible to the naked eye. For example, a human hair is between 50 and 70 micrometers, and a human red blood cell is 8 micrometers. Luminex's standard microsphere is 5.6 micrometers. At this size, the beads will stay suspended in liquid.
Microspheres can be made out of several different materials. However, Luminex uses beads composed of a polymer called polystyrene. This material has specific properties that make Luminex technologies possible.
The surface of the microspheres are covered with carboxyl groups. Biological agents, such as proteins and nucleic acids, can react or couple with these carboxyl groups to attach them to the microsphere. This makes it possible to run reactions on the surface of the microsphere by allowing proteins, antigens, antibodies, and nucleic acids to bind to the microspheres via the coupled biological agents.
Another property is the ability to introduce superparamagnetic particles to the polystyrene bead. Luminex utilizes both standard polystyrene beads that are 5.6 micrometers, as well as the magnetic polystyrene beads that are 6.5 micrometers. We call them Microplex and Magplex beads. The Magplex beads are slightly larger due to the added magnetic particles.
Another important property of the microspheres is the ability to be dyed. The first step to dye a bead is to prepare a solution of dyes in an organic solvent. A specific ratio of 2 dyes is added, red and near infrared. The beads are then introduced to the solution, represented by a single microsphere here. The beads will swell, absorbing the dye. When ready, the beads are removed from the organic solvent and moved into an aqueous solution. In this solution, the microspheres will shrink back to their normal size, trapping the unique dye signature within the bead. This process can also be done with an additional dye, giving the bead 3 dye signatures instead of 2.
The dye in the bead allows for identification. This set of beads has a concentration of dye increasing. Each bead has a greater concentration of dye from the last. Each bead can be identified by its unique concentration of dye. This bead has a D% concentration of dye 1. Adding another dye allows for a wider number of bead IDs. The bead with a D% concentration of dye 1 now has an additional value for the concentration of dye 2. This bead has a concentration of dye 1 at D% and dye 2 at H%. Adding a third dye gives our graph a third dimension, allowing for even greater number of bead IDs. This bead is identified as dye 1 D%, dye 2 H%, and dye 3 J%.
By taking microspheres with differentiating dye concentrations, a bead mix can be created to look for different possible causes of an illness. For example, if a patient has a digestive illness, a single test can be run using this bead mix, looking for 3 different illnesses, Salmonella, norovirus, or Giardia, in a single sample. This is called multiplexing. Looks like our patient has Salmonella.
The instrument analyzes the sample, and bead IDs are plotted into bead regions on a bead map for classification. Whether 2D or 3D, this bead map shows which beads are present.
Microspheres are an important part of Luminex's library of assay and reagent offerings. Their special properties allow for multiplexing in a flexible way that maximizes the information that can be gathered in a single sample.
XMAP technology is a multiplex
MagPix is a compact fluorescent-based detection system capable of simultaneously measuring up to 50 analytes in a single microtiter well. Unlike the Luminex 200 and FlexMap 3D systems, which are based on the principle of flow cytometry, the MagPix system is a fluorescent imager. The system consists of the following components. The system fluidics include the syringe pump, sample probe, and sample valve. These components are accessible through the side access door panel for ease of maintenance. The sample tray is compatible with standard 96-well flat bottom, V-bottom, and round bottom plates. Onboard dry fluid and waste containers include liquid level sensors to alert the user when the system is running dry and to prevent spillage.
Bead sets are combined into a single well to measure multiple analytes from one sample. Analytes
The heart of the system is the optics module, which includes the sample chamber, magnet, light-emitting diodes, and a CCD
and
camera.
finally, a reporter monitor.
Based on Luminex's proven XMAT technology, the MagPix system uses color-coded magnetic microspheres as the substrate on which assays are performed. MagPix can perform quantitative analysis of proteins and nucleic acids in a variety of sample matrices. Here's how it's done. The surfaces of Luminex Magplex microspheres are easily coupled with a reagent that is specific to the analyte being tested, such as a monoclonal antibody, protein, or DNA sequence.
In CCD imaging,
When the coupled microspheres are loaded onto a microtiter plate and mixed with the biological sample, analytes are captured based on specific antibody protein recognition or DNA hybridization.
excites and quantifies the assay.
The complex is biotinylated, and finally the detection molecule is attached.
Readings for each bead set and produces
The samples are now ready to be read on the MagPix system. A sample probe draws the suspended microsphere-based assay into the instrument.
Within a single sample, rapidly and accurately.
The beads are carried by the system dry fluid into a chamber.
XMAP systems are unique in the ability to provide both high throughput and high content data, making them ideally
As this is happening, a magnet moves into place to hold the microspheres in position. The microspheres settle in a monolayer fashion on the back of the chamber. Red light-emitting diodes, LEDs, illuminate the chamber, and the CCD camera captures a set of images of the fluorescent microspheres. The software processes the images and identifies the beads based on their fluorescent pattern. Green LEDs then illuminate the chamber, and if the analyte of interest is present, the CCD camera will capture an image of the fluorescent detection molecule. The software processes the image and determines the concentration of the analyte of interest from a standard curve. Once the sample has been read, the magnet is moved away from the chamber, allowing the microspheres to be swept out. The system then flushes the chamber in preparation for the next sample. Luminex Exponent software reports the results of the multiplexed assays in real time. In this way, MagPix allows the completion of up to 4,800 individual tests in less than 1 hour. MagPix is a cost-effective, versatile multiplexing platform that provides the researcher a tool that requires less sample, less reagent, and lower labor inputs than traditional testing methods such as ELISA, thereby reducing the cost per data point for testing of multiple analytes.
Designs, manufactures, and sells its own instruments. Let's take a closer look at each one. Luminex released the Luminex 100 in 1999. It was Luminex's first instrument. Some hardware improvements were made to the Luminex 100, and in 2005, the Luminex 200 was released. The Luminex 200 can read both microplex as well as magplex beads. The multiplexing capability using magplex beads is 80, while the microplex beads can be detected in 100 bead regions. It can analyze a full 96-well plate in about 45 minutes. The beads are interrogated using flow cytometry. The next instrument released by Luminex was the FlexMap 3D in 2009. It can also read both microplex and magplex beads. The FlexMap 3D can identify up to 500 different bead regions,
is Luminex's most
either
advanced
96
multiplex
well or
analyzer.
384 well plates
Dynamic
can be
range
used with the
to
FlexMap
simultaneously
3D.
measure up to 500
The
genes or
FlexMap
proteins
3D
from a
uses
small
flow
sample volume,
cytometry
make it
to
a most
interrogate the
flexible
microspheres.
multiplexing
Other
platform.
candidate capture antibodies to
4
The
different
FlexMap
microspheres
3D is used in a
working
wide
together.
variety of
When
applications,
The
using
MagPix
together,
instrument was
force
released
transfer
in
extracellular protein
2010.
expression
testing of
profiling,
all 4
It can
candidates.
identify
Cell
up to
signaling
50
profiling,
Magplex
Determine
bead
the antibodies.
regions.
pathogen
The
detection,
MagPix
Determine the best
instrument
antibody
only
vaccine
reads
testing,
Magplex beads
gene expression,
because
FlexMap
magnets
assays
are an
are
integral
SNP
part
genotyping,
of the fluorescent
optimal
imaging
antibody
analysis
serology,
used in the
and
MagPix
Performance of the
antibody
instrument.
assays is then compared
It
to
can
that of the
analyze a
original
full
MagPlex
polystyrene
96
assay
well
beads are at
with
the
plate in
regard to
heart
about
of
signal
all
60
strength,
Luminex
minutes.
dynamic
multiplex
range, and
assays.
sensitivity.
These beads are
The
available
results
in a
show that
magnetic
similar
format
antibodies
The
can
Luminex
perform very
200
differently
and
under the same
automation
MagPix
conditions, and that
in the
several
FlexMap
antibodies
3D
can
with
instrument
precise
simultaneously
are
concentrations
each
use the
used
Luminex
for
XMAP
both
assay,
protein
giving
and
each
nucleic
bead
acid
set a
applications.
unique color
sensitivity and
code.
dynamic range.
The
Using
Aries
this process,
system
we have
is a
created
fully
500
integrated
unique
sample
The
to
main
answer
advantage of this
platform
technique
that
over
performs
biological
existing
sample
methods
reagents
like
specific to the
ELISA
analyte bead
extraction,
is that
testing
XMAP
can be
based
coupled to
real-time
the
digital
PCR
surface of
assays allow the
beads.
researchers
This
to test
flexibility
The Aries
allows you to
system,
perform a
released in
variety of different
2015,
tests,
test
including
is
nucleic
multiple
unique
acid
that it
antibodies
assays,
doesn't use
simultaneously
amino
beads for
to identify
acids,
the
detection.
best
receptor
antibody.
ligand
It
assays,
utilizes
and
real-time
enzymatic
PCR-based
assays
will be Erica
multiplexing
Lopez
technology,
and associate
Three
scientists
incorporating
basic
at
types of
Multiplex
amino
reporters that
acids
fluoresce
can be
at different
performed with
wavelengths.
XMAP technology. Capture
Aries
sandwich,
uses
indirect
fluorimeter
detection,
detection during
and competitive
PCR
inhibition.
cycling to read
Here is how
assay
a capture
results.
sandwich assay is performed.
Aries can run 12
In this type of
steps
assay,
using
a
2
monoclonal
magazines.
antibody is coupled to the surface of the Luminex beads.
The Aries
When the
M1
coupled beads are
system
loaded
is a
onto a
fully
microtiter
integrated
plate and
sample
mixed with
to
a
answer
biological
platform
sample,
that
the
performs
protein
sample
of interest is
preparation,
captured based on
extraction,
specific
and
antibody
real-time
protein
PCR
recognition.
assays.
The
The
complex
Aries
is
M1
bioaccumulated
system
and
uses
finally the
fluorimeter
detection molecule is
detection
attached.
during PCR
The
cycling
samples are
to
now
read
ready to be
assay
read on the
results.
FlexMap 3D system.
It utilizes real-time
A sample probe
PCR-based
draws the suspended
multiplexing
feed of the
technology,
captured and labeled
incorporating
analyte into the
reporters
instrument.
that fluoresce
The
at
beads are
different
carried with a
wavelengths.
system sheath fluid into a cuvette where
The
the
system
randomly distributed
contains one
beads
module
line up in a
with
single
the
file.
ability to run
This
1
allows
to
the
6
particles to be
different
measured
assays
discretely.
that all use the Aries
As each bead
universal
passes through
protocol.
the cuvette, a red laser excites the internal dyes, allowing proper
The
classification of
Verigene
the bead,
system
which in turn
instrumentation
identifies the
consists
analyte
of a
attached to the
Verigene
bead.
reader and one
A
or
green laser
multiple
excites the labeling
Verigene
molecule bound to
processor
the analyte on the
SP.
bead, providing
The Verigene
information on
processor
the amount
SP
of analyte.
is a benchtop
Both
instrument that
classification and
combines
reporting
automated
are made on
nucleic
each
acid
individual
extraction,
bead, so the FlexMap 3D
purification,
system is able to precisely
amplification, or
determine multiplex
fragmentation,
assay results.
and hybridization in each module.
Luminex Exponent
The
software
Verigene
processes the
reader
data
serves as
and
the
can be
computer
used to
by
analyze the
managing
test
sample
results.
information and
Results
reading
obtained with
results from
Luminex software
processed
can also be
particles.
exported to any data
The
analysis
Verigene
program
processor
of choosing.
SP
Add
utilizes
500
The
nanogrid
FlexMap 3D
technology,
system assures
a
high
gold
throughput without
nanoparticle
compromising
chemistry,
flexibility or
to
to allow
performance.
for detection.
Optimize your workflow with
The
automated
Verigene
routine
reader
tasks,
utilizes
integrated front-end
nanoparticle
sample
imaging
preparation,
to detect
and acquire
target
more answers
presence
per
or
test than
absence.
ever before.
Covered after the coupling reaction using a
FlexMap
Up to
3D helps
32
you meet the
Verigene
constant demands
processor
for
SP
high-quality
can
data
be
with
connected
features
to
like
one Verigene reader,
fast read
meaning
times,
up
the
to
ability to
32
simultaneously
Verigene assays
measure
can
up
be
to
run at the
500
same
genes or
time.
proteins, a large menu of commercial kits and reagents to develop
Luminex
custom assays,
designs,
front-end
manufactures,
automation and
and sells
laboratory
various
information systems
instruments
integration,
for protein and nucleic acid
compatibility with both
applications.
96 and 384 well plate
4
formats,
micrograms per milliliter
automated
assay.
startup and shutdown routines,
Aliquot 50
and it's
microliters
easy
of
to
each test
use.
solution into 4 wells of a round
Hi,
bottom
my name is
6
Lyndon.
well
I'm a
plate.
technical
The total
support
is
agent here at
60
Luminex. I wanted to go over how to clean and set the height of a probe on
Then
a
add 50
Luminex 200
microliters of
instrument.
assay buffer
There are
into
several
the wells
reasons
to
that the
measure
probe may
background,
need to be
and
cleaned
50
or the height
microliters of
adjusted.
diluted confirmation
Whatever the
antibody
reason,
into
here's how to
well, the
is a
beads on
multiplexing
solution.
technology
Okay, folks, so
that
based on
generates
that,
more
you should
data from a
appreciate
single
the
sample,
importance
which
of
custom
the
methods through on the front
different
architectural
detection
design enables
systems
large
Washing
numbers
well
of
that can
bioassays
be
that
used
can
for
download
the
and
Nexus
analyze
approach
the
to
compounds
multiplex.
cost-effectively
From the
and
plate in a
accurately.
similar
That
fashion
finishes
using
FlexMap
multiplex.
technology
The main
allows
two
its color
certainly
speeds
that,
to
that
carry
we
out
Please
biological
be careful
assays
not to
are
damage the
similar to
sample
ELISA
probe,
MSD
or
such as
platform.
slide out
standard
easily.
acid
If you feel
optimization.
resistance when trying to remove the sample probe,
With
contact
precise
Luminex
concentrations
Technical
of
Support.
specific